Hello, everybody. Thanks very much for attending the webinar today. I'm very excited to introduce you to the BioFlux system and to talk about how it can be used to facilitate your platelet adhesion and aggregation assays. Let's get started with the agenda. And also, if you can't hear anything or you're having trouble, just please populate the chat box and we'll either slow down or talk louder or adjust any other parameters. Thanks. Today we're going to start with a quick background of Fluxion Biosciences, who we are, followed by a discussion of some of the parameters that are important for platelet assays. We'll then explain the BioFlux system and how it works, and finally we'll look at some applications of the system for platelet and whole blood assays, and finally at the very end we'll look at what the system configuration entails. Fluxion is a small company based in the San Francisco Bay Area. Our main focus is to design instruments and consumables to enable functional assays with live cells. Currently, we offer products in two main application spaces. One, for many types of live cell assays using microscopy, and that's the BioFlux. That's what we're going to talk about today. And two, for screening ion channel response to compounds, and that's called BioFlux, which we would be very happy to answer questions about or provide more information by request at the end. Our first system, the BioFlux 200, was launched in 2008. Since then, Fluxion's products have been adopted by labs worldwide for both academic research and for R&D in the pharmaceutical and biotech industries. What BioFlux offers and what we're going to talk about today is an added dimension or added dimensions of control over several critical parameters that influence cardiovascular biology and that are not all controlled for in a single traditional platelet function assay. Some of the controllable variables delivered by the BioFlux are shear, the importance of which we'll discuss in the following slides, maintenance of consistent conditions, for example, compound delivery or environmental conditions like temperature and atmosphere, as well as the ability to perform these in parallel and to collect dynamic data in time-lapse, which is especially important for the assays we're going to talk about today. All of these parameters can potentially enhance your understanding of the dynamic events occurring during platelet adhesion and aggregation. The first parameter we're going to talk about is shear. Shear is a mechanical force, in this case exacted by fluid flow, or I guess in this case really blood flow, in a healthy individual in the circulatory system. Shear can range from less than 1 dyne to 20 dynes per centimeter squared. In the case of a stenosed artery or other disease state, shear can be well in excess of 20 to hundreds of dynes per centimeter square. This ever-present force has a large influence on biological processes, causing changes in gene expression and cellular morphology and response to injury and many other processes. So it makes sense that shear should be a controlled parameter in many experiments, including those in cardiovascular research, stem cell biology, microbiology, and inflammation. Some approaches to study platelets, such as light transmission agrometry, do not provide controlled laminar flow or shear. And so in the next slide, I just have an example of how shear can contribute to both good and bad changes based on the situation. So if you look at these cross-sections of blood vessels, when you have normal blood flow, this is in hemostasis, you see the blood cells are flowing through this valve and there are no problems. And this is how we like to maintain our health and all that. If you look at the blood vessel on the far right, you can see that there is an atherosclerotic plaque formed within the vessel and it's occluding it. And when this happens, you have very high shear in the vessel based on how occluded it is, and this can contribute to spontaneous clot formation and leading to very, very pathological consequences. Here is another good example of where shear plays an important role in vivo. The biology of the endothelium is always under blood flow. When endothelial cells are grown under shear flow, there are changes in cell signaling leading to morphological and functional changes in cell behavior. In some cases, as I said before, the response to shear can be beneficial, such as resistance to injury, and in some cases, shear can contribute to arthrosclerosis and thrombus formation. The main application we're going to talk about today is the study of platelets under flow, and this is absolutely one of the best examples of biology directly affected by shear and force. In fact, the biochemistry and mechanobiology of platelet aggregation has a complex dependence on shear. At low shear rates, activated platelets aggregate by fibrinogen and GP2B3A. As shear increases, molecules involved in aggregation change to include a combination of von Willebrand factor, fibrinogen, fibronectin, and finally, at very high shear, von Willebrand factor alone. Therefore, in order to evaluate potential antithrombotic therapeutics as well as other aspects of platelet biology, a wide variety of subsurface and controlled shear conditions must always be tested. Just to finish up talking about some of the other BioFlux controllable parameters, besides shear stress, including flow and biological assays confers a number of additional benefits, such as full management of the microenvironment that the cells see, which is important to ensure consistent cell behavior time after time. As well, this provides the opportunity to change conditions reliably for other types of experiments. For example, to create areas of continuous increased chemoattractant in a chemotaxis assay, and to provide pulsatile flow to understand the role of cyclic waveforms on cell biology, which we'll talk about later. How does the BioFlux deliver these controllable parameters? Well, it was designed exactly with multi-parametric microenvironmental control in mind. Using the hardware, which we'll talk about shortly, and the microfluidic BioFlux plates, we can tightly control parameters of shear from 0 to 200 dynes per centimeter square, parameters of temperature from ambient to physiological, and use of external gas input from 5% CO2 to 100% nitrogen to buffer media or provide anoxic conditions for hypoxia models. Our unique fluid control interface allows the channels to be compartmentalized so that assays can be run under several conditions at once. In addition to controllable environmental parameters, the BioFlux was designed to be easy to use out of the box. The consumables are in a well plate format, and we'll talk about this in the next couple of slides. And as such, many assays can be run in parallel. In general, up to 24 simultaneously for platelets. In other types of assays, you can run up to 96 at once. And very important for blood assays, the devices are disposable and easy to clean up and set up. And certainly because we're using a microfluidic format, which by nature uses small amounts of reagents, this opens up the possibility of using precious clinical samples, such as pediatric draws, and rare cells to their full potential in platelet biology assays, as well as other types of assays. At the heart of the BioFlux system is the pneumatic controller, which affords very, very tight control over fluid handling and shear, and that's shown in the upper left panel. The controller couples to the well plate device via an airtight interface, and you can see that in the bottom right-hand corner of the controller diagram. Then data can be collected using microscopy, either manual or automated, and you can see down below on the lower left that we have a microscope stage. And we have made user-friendly software which enables both seamless data collection and concurrent flow protocols and automated data analysis. So you can see a screenshot of that in the lower right. So what is a well plate microfluidic? We have a movie clip, which is, while it's loading, it'll take a few minutes to load. I'll give you a little preview of it. In this video, you will see our devices, which consist of SBS well plates. The well plate serves as a chassis for the microfluidic channel. The plate will then turn over, and underneath, you'll be able to see the microfluidic channels. Those will be apparent. They run from well to well in independent units, and the channels are repeated in an array over the entire footprint of the well plate. At the bottom of each plate is a cover slip glass, and that's optimized for using an inverted microscope and microscopy imaging. So now the movie is just about loaded, and I will play it for you. So here you can see the well plate, and this is the top where you would actually pipette liquids into it. And now it's going to flip over, and you can see these channels that run from well to well in the plate, and it's now highlighted in yellow. And now you can see that there's an array of channels, all independent experimental units across the entire plate. So that just gives you an idea of how the device looks and how you use it and how the channels are arrayed. Okay, so the next video is an explanation of how a typical experiment works in the BioFlux plate. So as it's loading, I can just give you another quick preview. Coatings and cells are dispensed into the wells of the plate just as if it was a normal well plate. And then the interface is placed on the top, making an airtight seal. We use pneumatic pressure, which we apply through the interface, and then that pushes the fluids or cells into the channels, and then the cells of interest, such as blood cells, circulating blood cells, platelets, etc., can then be added, and you can assay for your effect of interest. So this is just about loaded, and I will play. Okay, so you can see here the cross-section of the well plate and pipetting some cells into it. You can see as you apply pressure, the blue fluid and the cells are moving into the channel, and now they're moving into the waste well. If desired, you can start a monolayer, and that's what's shown here, and then you can add your cells of interest, whether they're platelets or white blood cells, and then flow them through under controlled shear. So that just gives you a quick preview of how an experiment looks, and you would be observing it through the bottom of this plate here, so from the very lower part through the top. Okay, so each channel for all intents and purposes is a flow cell chamber, but it doesn't carry the normal baggage or fluid volume associated with a parallel plate flow chamber. The BioFlux plate serves as a self-contained array of flow cells with one controller instead of many, and very, very low volumes of media consumed. So instead of 24 separate flow cell setups that you would need to do 24 assays, you're just using one well plate and one controller for that. Here are some of the features that make the BioFlux plate conducive to live platelet assays. Number one, the well plate format makes for easy lab handling. Everybody knows how to use a well plate and pipette in and out of it just as you normally would any other type of tissue culture or well plate vessel. The cover glass at the bottom is optimized for imaging, so you can get very, very nice epifluorescence, confocal, and other types of imaging. The channels are all identical to each other, so this really favors experimental repeatability, so you expect the same experiment every time, and that's what you're able to do. And the design favors simplicity, so there's no liquids inside the tubes of the controller, so you don't have any contamination or cleanup. And the experiments themselves are self-contained within the channel, within the plate, so there's no setup overhead or cleanup overhead associated with a normal flow cell. Okay, let's quickly talk about the BioFlux well plate microfluidic devices. They come in two main formats, 24 and 48 well, with slightly different uses for each. The 24 well design shown here was engineered for experimental flexibility. So in this case, there are 8 independent channels, and each channel has 2 inlets and 1 outlet. What this enables is compound switching on the fly, as well as compartmentalization of the channels using parallel flow. This design is currently intended for physiological shear. In contrast, there is a 48 well format as well, and the 48 well format plates come in 2 shear ranges, which enable both physiological shear or lower flow rates and pathological shear or higher flow rates, which is very much of interest to the platelet biology space. Each channel is formed by 2 wells, 1 input and 1 output. This enables higher throughput per plate, more data points, etc. And you can see the channel design right and how it is arrayed over the plate on the lower left panel. The experiments we're going to talk about today exclusively leverage these plate designs, meaning the 48 well plate. However, there is utility for the 24-well design in platelet studies, either for washing or compound addition on the fly, or as the 24-well plate would suggest, longer run times. So there is some utility there. BioFlux applications run the gamut from the cardiovascular space to microbiology. However, today we're just going to focus on platelet biology, and so we're going to talk about platelet adhesion and aggregation on ligand, as well as some endothelial cell biology. And then at the very end, I'm going to talk about one of the newer functions of the BioFlux, which is pulsatile flow. We've applied this specifically to endothelial cell biology, but it certainly is extensible to platelet studies and platelet function analysis. Please let us know in the chat box if you have any questions about the assays that we aren't going to talk about today. We're certainly happy to share information about that. Okay, so some of the assays that have been validated in the BioFlux system for platelets are platelet adhesion to purified substrates such as von Willebrand factor or collagen, culture of endothelial cells for platelet adhesion, studies of platelet aggregation kinetics, as well as study of interactions between platelets and other cell types like white blood cells, etc. Just to whet your appetite, here you see a micrograph of one of the BioFlux channels after we perfuse whole blood over collagen. And so now we've perfused PBS and we're starting to dilute the erythrocytes, and you can see what this is leaving behind are some thrombi made up of platelets and some thrombi that are made up of both platelets and erythrocytes. And this was done under normal physiological shear at 10 dynes per centimeter squared. Okay. So analysis of platelet function under flow is requisite to understanding the complex biological relationships leading to hemostasis or pathology in the cardiovascular niche. The function of platelet receptors and the eventual biological outcome are strongly influenced by shear, which we talked about earlier, created by the flow of blood within the circulatory system. Many in vitro methods have been developed over time to be used in research labs to study platelet biology under conditions of shear flow. These include light transmission agrometers, cone and plate analyzers, perfusion chambers, and more recently microfluidic devices like the BioFlux. The devices listed on this slide all have certain limitations that either influence experimental parameters, such as the LTA, which does deliver flow but is not physiological or laminar flow, or reduced experimental flexibility like the PSA 100, which has set coding so you are limited in your flexibility, or model system choice. So when you consider the parallel plate flow chamber, this is one of the most flexible and physiologically relevant devices, but it requires extremely large volumes of blood relatively per experiment, which largely precludes murine or pediatric or even multi-parametric studies on the same draw. The BioFlux system itself occupies a unique niche among these technologies with benefits in many of the areas that are listed. First, it has much higher throughput and experimental flexibility and can fit into many types of experiments within the drug discovery pipeline. Some of the most important qualities of the BioFlux that differentiate it from other technologies are the ability to close in on in vivo models using laminar flow and or pulsatile flow, flexibility of substrate, use of human cells in very small blood volumes, as well as operation at very, very wide range of shears, both pathological and physiological. I just want to talk about this quickly before we delve into the applications proper, but one very important aspect of the BioFlux is blood volume per experiment, and it didn't really fit into the category of an application, so I just wanted to show you these data. Shown at the very left of this chart are selected shear stress and shear rates that would be pertinent to platelet assays. And shown on the right side of the chart are the volumes required per experiment. I should note that you need a minimum around 25 to 50 microliters to load the chip, but the volumes that are shown are what actually pass through the experimental region during the course of a typical 5-minute platelet adhesion assay. So on the very low end, where you have shear of 1 dyne, you're only using about 2 microliters of blood per 5-minute experiment. And at the very opposite end of the range, where you're doing a shear of 200 dynes per centimeter squared, you have only 300 microliters of blood consumed, which is one order or orders of magnitude less than you would in a parallel plate flow chamber. So this is a very important aspect of the BioFlux that differentiates it from other technologies. Let's now talk about some of the applications we've tested using whole blood and platelets in the BioFlux system. One very important aspect of many platelet assays is the capture of dynamic data, meaning data capture in time-lapse, so that all the behavior that occurs during thrombus formation or any other biological assay is captured. One interesting behavior that can be seen with platelets is rolling on substrate, and changes in morphology of the platelets themselves as they're rolling, which is all dependent on shear. The BioFlux is well suited to capturing this type of data, as well as providing the tools for data analysis. Typically, rolling data can be used to understand or to characterize receptor-ligand interactions in response to inhibitors or other variables like different cell types or transgenic cells or mutants or whatnot. What you can see at the top of the slide is a micrograph, which is stacked over 30 frames. In this case, these are white blood cells that appear as a white streak. These cells are actually rolling and slowing down on the substrate, which in this case is VCAM, and the distance versus time can then be measured to determine the velocity using BioFlux software. Here are some still images as well as a stacked set of images for platelet rolling behavior. That's shown on the upper right. This experiment is on purified human von Willebrand factor at 10 dynes per centimeter squared. And from the micrograph shown on the upper right-hand side, you can determine the velocity of platelet particle movement by measuring the trail left by the rolling platelet, which could later be compared to other substrates or drug or buffering conditions. And I do want to mention that we have movies of most of the experiments that we're talking about today, but because the downloads are very slow for the webinar, I didn't include them all. So if you're interested in any of these movies, we're very happy to provide them to you by email after the presentation. So just let us know in the chat box. Let's continue our application discussion with platelet adhesion and aggregation on purified ligands. The experimental setup here involves coating the BioFlux channels with a purified ligand and then perfusion of whole blood or purified platelets over the coating. On the right, you see snapshots of typical platelet adhesion assays under flow on very commonly used substrates. The top panel shows what we consider reversible platelet adhesion on von Willebrand factor. Under physiological conditions, platelets will typically roll on the substrate, forming a single moving layer of particles. In contrast, in this case, under these conditions, on both fibronectin and collagen, platelets can adhere to the substrate and will subsequently aggregate on top of each other, forming thrombi. Once an adhesion model is established in this way, the same type of experiment can be used to generate compound response data, such as the IC50 data for GP2B3A inhibitors, which are shown on left. Here we're testing the effect of shear on dose response with platelets on collagen. The top plot is at 10 dynes per centimeter squared, and the bottom plot is at 20 dynes per centimeter squared. What you can see is that the dose response shifts as the shear increases. Also, please note that these experiments can all be performed with the same blood draw at the same time, so that variability is eliminated due to multiple draws or a draw sitting around prior to execution of the experiment. So where the BioFlux is very useful in this way is that you could take one human or mouse blood draw and use the entire draw in the same plate at the same time under the exact same conditions and really eliminate that variability due to draw time. A very unique capability of the BioFlux system, most notably the BioFlux 1000, is the ability to perform platelet adhesion and platelet aggregation studies in parallel channels simultaneously, as I just alluded to. And we use automated microscopy to do this, and also the parallel nature of the plate to enable it. Besides the obvious advantage, which is increasing throughput, this gives the first time the ability to use the same donor for multiple conditions. This ultimately eliminates unwanted variables on the single data set due to different donors or blood aging in the presence of anticoagulation treatment. Shown here, this is an assay that we did with sodium citrate anticoagulated human whole blood on human von Willebrand factor at 125 dynes per centimeter squared. This is in the medium to high pathological shear range. Interactions occurring at this shear are primarily governed by von Willebrand factor as the substrate and by the GP1b5-9 complex. The blood here was labeled with calcine AM, which is known to stain the platelets but not erythrocytes. Shown here are time-lapse data from a total of 18 points, or several channels of a BioFlux plate over a 10-minute time-lapse. What you can see is that not only can we collect data during a thrombosis time-lapse in many channels, but we get good reproducibility from many points shown on the plot, making this type of assay a very good base for a screening experiment. Shown here is a montaged version of this experiment where you can see each panel of the time-lapse. So it's a montaged image of one of the data capture sets during the application of shear, where each panel is a 30-second interval for a total of 10 minutes. You can see the thrombus formation over time on von Willebrand. These data are very useful, for instance, when you're comparing the platelet behavior among parental and knockout mice to determine if there's a kinetic effect related to a particular change in a receptor-ligand interaction or signaling pathway normally leading to thrombosis. The BioFlux system is especially suited to culturing mammalian cells inside the channels to be used either as a substrate for an experiment or as part of an experiment itself. Endothelial cells are often grown in the BioFlux for such studies as white blood cell adhesion under flow, transmigration, and cellular response to shear, as well as many other types of assays. And I should also note that we have grown many other cell types within the channels as well. Endothelial cells are especially well suited to growing under flow because they naturally grow under flow in vivo. So we've had quite a lot of experience with these cells. So we've used endothelial cell monolayers in the next few slides to test the effect of a compound, we'll call it compound X for now, on platelet adhesion to monolayers. Shown here are just a few examples of different types of platelet interaction with endothelial cells. So if you look at the top panel on the right, that's just an endothelial cell monolayer with no platelets, no blood added. In the middle panel, what you can see are, well, you can see a massive thrombus that's formed in one part of the channel, but there's also several small thrombi around it. So that's one outcome of an experiment on a stimulated monolayer. And then below, on the very bottom panel, you can actually see this is a platelet adhesion assay, and every single one of the endothelial cells are completely coated with platelets. So you can look at many different types of assays on endothelial cells. And one nice thing to note here is that you can use both human endothelial cells and human blood to recapitulate an in vivo scenario ex vivo. Okay, shown here is a preliminary experiment where a compound of interest was applied to the endothelial cell monolayer themselves inside the BioFlux channels, and in this case, this was over set durations. Then after that was completed, the whole blood was added under shear flow, in this case in the physiological range at around 20 dynes per centimeter squared. Platelet adhesion data were captured under flow, and we found that both the dose and the duration of treatment had measurable effects on platelet adhesion. Shown here are some of the representative data where we measured platelet adhesion as well as cell density in the monolayer using nuclear stains. Green, what you see as fluorescent green, are the adherent and streaming platelets, and blue are the nuclei of the endothelial cells. If you just focus on the top two micrographs, you can see that on the left there are cells that have become completely coated with adherent platelets, and on the right, the mock-treated cells have no adherent platelets at all. And you can also see, as I said, there's green streaks, and those are just platelets flying by, and we don't measure those. Shown here is how we actually quantitate the data. We threshold the fluorescent signal only from the adherent platelets, not platelets that appear as the fluorescent streaks. So what we're actually measuring is shown in orange. To count the endothelial cells, we measure the number of nuclei in the DAPI channel, and we can then express the adhesion data in the context of cell number, or they can be expressed by themselves. So that just gives you an idea of both platelet adhesion assays on ligand and platelet adhesion assays on cells. Let's switch gears, and I just want to talk about one of the newest features of the BioFlux system, which is the ability to produce cyclic waveforms, which adds additional relevance to experiments in the cardiovascular niche. I'm not going to talk about any platelet assays here specifically, as I said at the beginning of the webinar. However, this is yet another variable that is present in the cardiovascular niche and that should be accounted for in certain experiments. One example I am going to talk about today is the effect of waveforms on endothelial cell behavior. In particular, the propensity for certain waveforms to protect against atherosclerotic plaques and waveforms that actually contribute to plaque formation. Shown at the top are two such waveforms that were actually measured from the carotid artery. You can see the protective waveform on the left and the prone waveform on the right, which means it's prone to forming plaques. Below, there's a schematic of the carotid artery where it's branching and where these two waveforms are found. So the protective is found higher up on the branch, and the prone is found at the branch itself. Shown at the right are sinusoidal waveforms generated within the BioFlux system, both at 0.5 hertz, and the amplitude is 20 dynes per centimeter square here. And then below you have 1.2 hertz, and this is equivalent to 72 beats per minute of pulse. And these are actual measured waveforms off of the BioFlux controller. To begin to understand the effects of variable or pulsatile shear stress on the cell junctional integrity and maintenance of endothelial cells, we subjected HUVEC monolayers to either continuous shear conditions, or C, at 0.02 dynes per centimeter square, which is essentially no shear, 5 to 0 dyne per centimeter square intermittent pulsatile flow at 1.2 hertz, and intermittent flow, P, with a maximum amplitude of 20 dynes per centimeter squared at 1.2 hertz for 24 hours, and that's the experiment that's shown here. Following application of the shear waveforms, cells were fixed immediately and stained by indirect immunofluorescence for connexin, and that's expressed at the cell junction as well as inside the cell. In this case, we assayed for expression of both connexin 37 and 43. All of the capture conditions that are shown here were identical, meaning we have the same exposure time on the camera, same experiment, same day. Interestingly, we found expression of connexin 43 was upregulated in the 20 dyne intermittent flow condition, and it was localized distinctly to the perinuclear area. So if you focus on the middle overlaid micrograph on the right, you can see the green fluorescence is connexin 43, and you can see that it's directly adjacent to the nuclei. In contrast, connexin 37 was upregulated in the 5 dyne pulsatile flow condition compared to both gravity flow and the 20 dyne pulsatile flow condition. So there you can see on the top right there is both red and green fluorescence, and the red is connexin 37 and the green is connexin 43. This is actually in contrast to what has been reported in the literature and really suggests that we need to study many conditions of pulsatile flow further to really understand what's going on in the prone and protective waveforms. So in this case, we measured raw fluorescence intensity at a set threshold, as I showed you before, so that orange thresholding, and expressed this as a function of the total cell number in the field. And you can see intensity was normalized to the no shear condition. So you can see that the connexin 37 is upregulated about twofold, and you can see connexin 43 in the 20 dynes is upregulated quite a bit as well. So you can actually measure what's happening inside the micrographs. Another aspect of this experiment is to follow the change in cell polarity and cytoskeleton during the application of variable shear. So we can also investigate this using the pulsatile flow functions of the BioFlux. Shown here are endothelial cells that were exposed to long-term, so 24 hours or longer, constant or variable shear at 1.2 hertz. The flow direction is from left to right. At the end point of the pulsatile shear experiment, cells were stained with phalloidin Alexa 488, which is going to stain the actin. There is a very marked difference in both the actin polarity as well as the monolayer density, and this is something that we certainly want to follow further. But if you focus on the merged micrographs in the green and blue row, you can see that under the continuous flow conditions, you have more or less a monolayer cells, and the cells are disorganized. There's no polarity to the actin. And then if you look at the variable shear, you can see that the cells are aligning to the flow from left to right direction. And in the case of the 20 dyne per centimeter square, the cells are actually enlarging with the pulsatile flow to cover the space. And this is certainly something that could be studied further. So hopefully you understand a little more about the parameters that are accounted for in a typical platelet assay and how important it is to include shear in the assessment of platelet biology. Also, I hope you've learned a little bit about the BioFlux and how it provides control and ease of use regarding many important aspects of your platelet assays, including physiological parameters and dynamic data capture, and how you can use just one system to perform many types of assays. That concludes our discussion of platelet applications in today's webinar. For your information, here are 3 product offerings for BioFlux. The 200 system consists of the controller, software, and accessories, everything needed to run your assays if you have your own inverted microscope, so that's shown at the top. And the BioFlux 1000 and BioFlux 1000Z. These are both fully automated and integrated systems that include automated image capture, automated hardware, and all the software needed to run walkaway automation and data analysis. If you have further interest in the system, would like more information about the system or applications, please visit flexionbio.com, or to inquire about ordering, please contact our sales department at sales@flexionbio.com or at 866-266-8380. Thank you very much for your attention today, and I'd be happy to answer any questions at this time. So I do see one question about calculating shear rate from dynes to centimeters squared to inverse seconds, and that's a very common question. Within the BioFlux software, this is actually a parameter that you would type in the shear that you desire. You would type in the viscosity for your solution, and then it would automatically calculate the inverse seconds. So let's see. Here's another question. Is collagen 1 fibrillar or a monomeric? We've used both fibrillar collagen from Helena and Chronolog, as well as sheared collagen from Invitrogen. It just depends on what you would like to assay. I don't see any more questions, so I would like to conclude our webinar. Thank you again for attending, and please let us know if you have any further questions. Have a good day.